Inverse PCR. Inverse PCR is a standard PCR in which the primers are oriented in reverse direction. The procedure of the inverse PCR or the characteristics of the inverse PCR are: in case of inverse PCR, template for the reverse primer usually is a restriction fragment that has been self-ligated. Inverse PCR function to clone sequences flanking to a known sequence. Flanking DNA sequences are digested and then ligated to generate a circular DNA. So in case of inverse PCR, usually the reverse primers are from restriction fragment which are self-ligated, and the flanking DNA sequences are digested and then ligated to generate circular DNA. Identification of sequences flanking transposable elements. In most of the cases, inverse PCR is used to identify those sequences which are flanking to the transposable elements. So inverse PCR is another type of the polymerase chain reaction in which we use PCR when only one internal sequence is known. This type of the polymerase chain reaction is usually useful in identifying the flanking sequences to various transposable elements or to various genomic inserts.